Let's go ahead and get started. Yes, afternoon, indeed. All right, so our goal today is to continue our discussion of the urinary system. We got the anatomy done and under our belt in the last class, and there was a fair amount of it, so hopefully you spent some time with that. And so now we can start talking about the physiology. And then if you have time, continue to work on your anatomy, study those models, study those charts, study those materials that you're going to be responsible for. We have one more lecture on Tuesday, 2 more assignments, so that should keep you very busy this weekend. This weekend is also your big weekend to study this material, so make sure you're looking at it and getting ahead. Because Thursday we have our lab and lecture exams. So again, we have the dreaded Thursday lab and lecture exam, which means 2 things. One, that we are getting new material 2 days before the exam, so make sure you're looking ahead of time so it's not fully new. You're prepared for that. But the other thing that means is we have 5 days off before we come back. And when we come back, we only have 4 lectures. Yep. So to encourage you to start looking ahead at the new material, I have not 1, but 2 assignments you are going to be responsible for. The first, as I usually do when we have a Thursday exam, you've got a pre-lab. Again, it's not the review. It's going to be the pre-lab for Unit 27 of the reproductive system. The other thing I'm going to have you do is one of the big things we're going to be doing on that Tuesday when we start is talking about meiosis and mitosis. And if you have forgotten or don't remember or never taught it properly, I want to make sure you look at mitosis and understand mitosis. So I'm making you do the lab start ahead of time as well so that you can be prepared. We will be talking about it together, but make sure you're comfortable with mitosis. And it wouldn't hurt to look at that meiosis. So do that activity and that will help us. Then we have a holiday that on Thursday, and then we come back 3 straight lectures and then lab and lecture exam final, and we are done. So it is rapidly approaching the end point. All right, questions on that? Oops, hold on 1 second. Apparently I forgot to end my office hours. All right, perfect. You guys are all still here though, right? I'll take that stunned silence as a yes. Excellent. Oh, some people did. Excellent. All right, perfect. Excellent. All right, and it looks like we are recording. All right, perfect. Excellent. So, any questions on the game plan? Yes, so the 24th, we do have a lecture. There's 2 assignments are going to be due. We do have a lecture, and we are going to be, like I said, we're going to be starting reproductive system. So yes, that's the other thing. We do have a lecture that week. We do have, it's 25% of the material we're going to be covering on this. With this section, it's got 4 lectures. So you want to make sure that you are absolutely there. 2 assignments that are due, but also absolutely we are doing a lecture on the 24th as well. Our days off are Thursday, Friday, and Saturday. So we do not have, and again, my Monday, Wednesday class, they don't have a day off. They have to lecture both days. So you're only getting 1. You're at least getting a holiday. But they had yesterday off, so that makes up for it. All right, great question. Any others?
I have a question about last lecture.
Yes.
So vasa recta, it's the 3rd capillary bed, right?
Correct. But the key to that is to remember that not every nephron has a vasa recta. Only the juxtamedullary nephrons, which only make up about 15% of the nephrons, have the vasa rectus.
So if it's short on the picture on the model, if it's short, it's peritubular capillary. If it's long until the pyramid, so it's a vasa recta, right?
That is correct. Yeah,
okay.
The cortical nephrons will have capillaries over their loops of Henle, but it will be just a peritubular capillary. Whereas the vasa recta capillary is only over the long juxtamedullary nephrons. And yes, Nick, absolutely. And typically the vasa rectas have more of a linear, more of a draping kind of appearance, whereas the peritubular are much more irregular. And absolutely, those juxtamedullary nephrons will we'll have a paratubular capillary around the proximal and distal convoluted tubules, and then we'll have a vasa recta on its loop of Henle.
One more question.
Yes.
So cortical nephrons, there are like 85% of that. It means they are more usual, or it's in one kidney, it's 85% of them and less of the juxtamedullary nephrons.
My understanding is the number of that you have on both sides is relatively identical. So yes, in both kidneys you have 85% cortical nephrons and 15% juxtamedullary in both. And they're intermixed too. It's not like you just have one segment where all of the juxtamedullary are together. They're going to be interspersed throughout.
Thank you.
Yep, great questions. Any others? All right, perfect. Then if we are good with our anatomy, then what that means is we can move on to our physiology. We at least started our introduction to the physiology in the last class. Put that there, perfect. Where the formation of urine, or really the filtering of the blood, is a three-step process. The first is the glomerular filtration, which again takes place only in the renal corpuscle. And remind me about this process. Is it passive or is it active?
This one passive.
Passive, excellent. What direction does it go? Into the blood or out of the blood? Out of the blood, excellent. This is where we make our filtrate, spectacular, right? Then we have tubular reabsorption. Where does that primarily occur?
In the proximal convoluted.
Excellent, proximal convoluted tubule. Is it active or passive?
Active.
Active. And what direction does it move?
Out of filtrate.
Out of the filtrate into the blood. Excellent. And then our third process, which is really 2B to tubular reabsorption's 2A, is tubular secretion. And again, we number it that way because where does most tubular secretion occur?
In the proximal tubule.
In the proximal convoluted tubule. Excellent. So again, they're both occurring at the same time. It's not like all the reabsorption occurs and then all the secretion occurs. They're all happening at the same time. What direction does tubular secretion go?
Into filtrate.
Into the filtrate, out of the blood. Excellent. And is it an active or a passive process? Active. Excellent. And again, that means that steps 2 and 3, tubular reabsorption, tubular secretion, can be selective. They can be specific about what they want to move, whereas filtration, being passive, can't be specific, right? It's just going to be basically based on size. So let's talk about that glomerular filtration. You're going to learn everything you ever wanted to know about glomerular filtration and more. So let's go ahead and start that process. Again, our filtrate is formed in the renal corpuscle, but as we've mentioned, filtrate is not the same thing as urine. How much filtrate did we say we produced during the course of the day? Yep, about 200 liters. Excellent. And how much urine do we produce during the course of the day? Yep, about 1 to 2 liters. Excellent. All right, so more than 99% of it is reabsorbed. Excellent. Basically, that filtrate that comes out is essentially the blood plasma. Water with a bunch of stuff dissolved in it. However, it's not identical to blood plasma because it's lacking some of the things that are in blood plasma. And those things are primarily the large things. There are going to be no formed elements, and there are going to be no large proteins. But pretty much everything else, both good and bad, because again, it's not selective, is going to be released with the water into our Bowman's capsule. Our goal then is going to be to reabsorb the vast majority of that. So what leaves our body as urine is primarily metabolic wastes and any unneeded substances. Great question. So Jessica asked, is it possible for there to be formed elements in the blood? there can be formed elements in the blood. You can get blood in the urine. You can get white blood cells in your urine and things along those lines. But usually those are an indication of some type of problem. Some of those problems could actually be outside the kidney, like for instance, a bladder or urinary tract infection. Ulcers or injuries can cause blood in there. But there are some cases where you can have formed elements in your urine that are not necessarily indicative of a problem. For instance, in some cases, extreme endurance athletes, because of the stress that they're putting on their bodies, can sometimes cause enough dilation of those glomeruli where you might get some formed elements. And actually pregnancy, it is not that uncommon for a pregnant woman to have formed elements in her urine. And again, it isn't always necessarily an indication of a problem, but of course you do want to check for infections and things along those lines as well. But yes, absolutely. In typical urine, there should not be any of those formed elements. All right, excellent. So, like I said, this is a passive process. We cannot be selective. And it is also going to rely then on pressures, primarily our blood pressure, to force our filtrate out of the glomerulus. Remember, one of the things we talked about the glomerulus is a very, very highly specialized capillary. And one of the ways it's highly specialized is that it has a high blood pressure. And that high blood pressure is going to play an important role in helping us to be able to regulate the movement. Just out of curiosity, does anybody remember what the typical blood pressure in a capillary was? I know we had an arterial end blood pressure and a venous end blood pressure. Does anybody remember either of those two values?
35 and 17, no?
There you go, absolutely. 35 millimeters of mercury at the arterial end and 17 millimeters of mercury at the venous end. Excellent. Absolutely spectacular. So absolutely. So we know our starting point. We kind of remember that. And let's stick this up in the corner to help us remember when we get to that point. And so as our blood passes through the glomerulus, this high pressure forces about 10% of the blood plasma out. And like I said, we produce around 200 liters of filtrate during the course of the day, but then have to reabsorb around 99% of that. Because this is a passive process, we want to do things that are going to maximize the efficiency of that filtration process. So we have a very thin, tiny membrane. We have a large surface area. And like I said, we are going to have that high glomerular blood pressure. These are numbers we are going to need to know. Now, we talked about the anatomy in the last class, but I want to do it again. Let me grab this again. I want to keep this here. So put that there for right now. Again, it lies between the glomerulus and the capsule. And as we talked about in the last class, it has three distinct layers to it. It has the fenestrated endothelium of the capillary. So that is, of course, a simple squamous epithelial tissue with fenestrations in it. That visceral layer of Bowman's capsule, which are those special podocytes that have those special foot-like processes. And then, as we talked about in the last class, it has the fused basement membranes for those together. In this case, those fused basement membranes are sometimes referred to as the dense layer. And I can show you why right here. Notice here we have an electron microscopy view. Using an electron microscope, we can actually see this filtration membrane. Here on the inside is our endothelium of our capillary. Here is those foot-like processes of our podocyte. And then here you can see the two fused membranes form this dense line along the center, and that is why it is called the dense layer. Let's get rid of that. As you can see, these openings in between them are tiny, only about 6 to 9 nanometers in diameter. So like I said, nothing big, no formed elements, no large proteins are going to be able to get out. But glucose, ions, ATP, amino acids, all the little stuff that is being transported by our blood plasma can all be expressed out of our glomerulus and into that Bowman's capsule. Now, we are basically looking at a capillary exchange. If I remember correctly, when we talked about capillary exchange before, there were 4 pressures that were involved in that process. And let's make this. What were the 4 pressures we identified that played a role in capillary exchange?
Blood pressure.
Excellent. The blood pressure, absolutely. Blood pressure was one of them. But what was the other way we referred to the blood pressure? Blood pressure, remember, is the force of the blood, or really the water of the blood, pushing against its boundaries. So we called that hydrostatic pressure. But you are 100% correct that the hydrostatic pressure of the blood is simply just a fancy way of saying blood pressure. Absolutely. What was another one of those?
Glomerular pressure.
True, absolutely. Or in this case, as osmotic pressure, right? So again, I see someone wrote here, several people wrote osmotic pressure of the blood. So our blood's still going to be here inside of the capillary, and it is still going to have stuff in it. And so that is going to be the osmotic pressure. What were the other ones? I thought someone had said it. There you go. Absolutely. We're going to have the hydrostatic pressure outside. Of that really close capsule that is wrapped around it. So that would be the hydrostatic pressure of Bowman's capsule. Right? If you were to take a hose and you were to put that hose inside of a cup, right, and turn the hose on, what would happen? Would you want to have your face right in front of it while you were doing that? No. All right, why not? When you, okay, let's take a more hopefully relatable example. You need to fill a bucket to wash your car. When you do that, do you take the hose, put it on full blast, and put it all the way down to the bottom of the bucket where it is an inch away from the bottom of the bucket? Is that how you're going to fill your bucket? The water starts at the bottom when you fill it. Why not approach it out of the bottom? Exactly. It'll splash up all over the place. When the water's coming out and that boundary is that close, it's going to bounce around. And so absolutely, we're going to have a hydrostatic pressure of that tight Bowman's capsule around it. What was the 4th pressure? When we were just doing normal capillary exchange. Well, it was osmotic pressure of the interstitial fluid, right? The water of the tissue surrounding the capillary had stuff in it, and that stuff had the potential to attract water to it. So when we were doing a normal capillary exchange, that was the 4th pressure. However, when our filtrate leaves the glomerulus into Bowman's capsule, does it stay in Bowman's capsule? No, it leaves Bowman's capsule and enters what?
Proximal tubule.
Proximal convoluted tubule, absolutely. So it's going to go to the proximal convoluted tubule. Because of this, there really is no osmotic pressure to the Bowman's capsule. So as a result of that, when we are trying to calculate our net filtration pressure, we don't have to worry about an osmotic pressure in our capsule because the fluid doesn't stay there long enough for it to have an effect. And so this is why we only have 3 pressures that we have to worry about. Now, let's figure one more thing out. As we learned before with this capillary exchange, these pressures can be outward or inward. Our hydrostatic pressure of the blood, is it an outward or an inward force? Out of the capillary or into the capillary? Outward pressure. What about the osmotic pressure of the blood in the capillary? Outward or inward? Excellent. And what about the hydrostatic pressure of the capillary? Outward or inward? Is it trying to pull water out of the capillary or is it trying to push water into the capillary? Inward. This is also an inward. So notice when calculating our net filtration pressure, we have to take our outward pressure. And from that, we have to subtract all the inward pressures. And so our net filtration pressure is going to be equal to the hydrostatic pressure of the capillary minus the osmotic pressure of the capillary plus the hydrostatic pressure of the capsule. Hydrostatic pressure of the, let's say the blood. So there's no confusion. Hydrostatic pressure of Bowman's capsule. We'll do Bowman like that. So we combine our inward forces, and that allows us to calculate what our net filtration pressure is going to be. So let's, oops, wrong button. Let's add some numbers to this. Remember, in a typical capillary, at the arterial end, the blood pressure was 35 millimeters of mercury, and at the venous end, it was 17. But notice here in our glomerulus, it is 55 millimeters of mercury, right? Much, much higher pressure than a normal tubulary capillary. Our capsular hydrostatic pressure, the Bowman's capsule hydrostatic pressure, is 15 millimeters of mercury. And what direction was that force again? The capsular hydrostatic pressure? Inward. Excellent. So this is in, this is out. And our blood colloid osmotic pressure is about 30 millimeters of mercury. And what direction was that one again? In or out? I've seen both now. Blood colloid osmotic pressure is the stuff in the blood. Is it bringing water to it, or is it pushing water away from it? Inward. This is an inward first, bringing it in. Excellent. So based on these numbers then, what would our net hydrostatic pressure equal? Pardon me, our net filtration pressure be? It would be exactly 55 minus 15 plus 30, which of course is 55 minus 45, and that equals a positive 10 millimeters of mercury outward. Bingo. And so these 3 pressures combined to give us an outward force of 10 millimeters of mercury. All right. Questions on that? All right, excellent. If you don't mind, there we go. If you don't like my art, here's the pretty picture that shows this. And so again, as we mentioned, anything smaller than around 9 nanometers and water is going to be forced out of the glomerulus. So I have a high blood pressure, hydrostatic pressure, osmotic pressure, pushing it back in, giving us a net filtration pressure of about 10 millimeters of mercury. And water too. All right. Questions on that? Now, this glomerular filtration rate, this net filtration pressure determines, so let's do that. Net filtration pressure determines the glomerular filtration rate. Absolutely, right? Because again, remember, our goal is to make that filtrate so we can filter the blood. If the net filtration pressure is not positive, is not an outward force, well, there would be nothing in the Bowman's capsule to draw back in, but we would have major issues. That would mean we wouldn't be able to clean the blood. And that would be a very bad thing. So our net filtration pressure determines our glomerular filtration rate. The glomerular filtration rate is the volume of filtrate that is produced per minute. Let's do this again. And those rates are about 125 milliliters per minute in males and about 105 milliliters per minute in females. And of course, why the big difference? That's right. Jessica mentioned it. It's because guys are better. No, again, primarily size. Absolutely. Yeah. And again, is that really a significant difference? No. All right. But this leads us back to kind of what we were talking about before. We want to maintain a consistent glomerular filtration rate. If our glomerular filtration rate is too high, if what that means we are making filtrate too rapidly, what is the effect of that going to be?
Not good reabsorption.
Right, exactly. So we're going to end up under processing the filtrate. We're not going to be able to process the filtrate fast enough and appropriately because it's going too fast. Conversely, if the glomerular filtration rate is too low, then what that means is we are making filtrate too slowly. And what happens then? Overprocess the filtrate.
May I ask you?
Yes.
I was thinking that it's not about the filtration rate if it's too high, or if it's under processed or over processed. I was thinking that it's more about the amount of water in the urine to go. Like, it's filtration rate, it's good, right? But it's not good if there are too much urine, right?
So great question. So it isn't so much when we talk about over and under processing the filtrate, we're really not necessarily talking about the amount of water. What we're really more talking about is the amount of stuff in the water. There are going to be ways that we control the water and that will regulate how much water is in our urine. But that's not what we're really talking about in this case. Maybe indirectly, because as we know, where solutes go, water follows. So for instance, if we are under processing the filtrate, there may be too many solutes in the urine. And if there's too many solutes in the urine, then more water may be lost with that. But that's more of an indirect issue as opposed to the direct one. Same thing if you're overprocessing. If you're overprocessing, too much stuff may be brought back. And if too much stuff is brought back, too much water may follow back as well. Again, it would make your water more concentrated, but at that point, that's not really the problem. The problem is more that you're overprocessing the filtrate, bringing things back that you don't want to bring back, and the effect that those would have on your body. So yes, volume could be influenced by this, but it's more indirectly, and it's more the substances in it that we're worried about. Okay. Now, Jessica also asked a question, again, controlling this glomerular filtration rate. Is this where the sympathetic nervous system comes in to play a role? Absolutely, it does. Our glomerular filtration rate has to be regulated very, very carefully, very, very constantly. And there are actually going to be 3 ways that we regulate it. One will indeed be from a long reflex involving our sympathetic nervous system. But guess what the other 2 ways are going to be?
Regulating of blood pressure?
Yeah. Well, okay. So, and we can do that systemically, but we can also do it locally. So we're going to use long reflexes, we're going to use short reflexes, or intrinsic, so extrinsic and intrinsic neural mechanisms. And what would the 3rd way be?
Nephrons, hormones can regulate it, right?
Yeah, there you go. Hormones, absolutely, hormonally. So that will be the way that those are going to be the 3 ways that we are going to regulate this process. And we need to talk about all of those. And there you go. Jessica, perfect lead into what we're going to talk about yet. Next, we need to talk about controlling the glomerular filtration rate. And there are 3 main mechanisms we are going to use for regulating this. All right. We're going to talk first about our neural mechanisms. And again, there are going to be 2. One intrinsic and one extrinsic. But we need to talk a little bit of vocabulary is not quite the right word. I'm not sure. I don't know what the right word is. We have to talk about some rules. I think this is probably the best way to describe this. Our goal is going to be to control blood pressure primarily. Yes, as we also talked about, we can control the surface area, for instance, of the glomerulus, and that can affect the filtration rate, and we will talk about that as well. But let's talk about controlling blood pressure, right? And that blood pressure is also going to be related to the flow. Are going to be related. Oops, I spelled related correctly. So let's think about what happens when we constrict a blood vessel. When we constrict a blood vessel, there are basically going to be 2 potential effects. What are those 2 potential effects going to be? Excellent. So one of the things that will happen is we will decrease blood flow through it. And what's the other? Perfect. And there's the other one as well. It can also potentially increase blood pressure. So those are, when we constrict a blood vessel, one of those two effects are going to occur. Conversely, when we dilate a blood vessel, right, there's going to be two potential effects. And those two potential effects are going to be to do the opposite. Increase blood flow through the vessel. And the other thing that it can do is it can decrease blood pressure. All right. So far, so good. All right, excellent. So let's go back to our lecture now that we've kind of thought about this a little bit, and we can talk about how these rules are going to help us to understand these mechanisms. It can potentially affect blood pressure, yes. All right. The first intrinsic mechanism is our renal autoregulation. Again, as we've talked about, we have all of these individual nephrons, and these individual nephrons need to monitor themselves. So this is our first concern, that autoregulation, that intrinsic activity. And there are two ways that it does it. The first way it does it is actually what we call the myogenic mechanism. Myo usually refers to what?
Muscles.
Muscle, absolutely. And in this case, in particular, we are talking about the smooth muscle of the afferent arteriole. Okay. Now, our goal with this particular mechanism is to maintain consistent pressure in the glomerulus. And the problem with this is that your blood pressure, is it consistent throughout the day? No. In fact, when you go from a sitting to a standing, or from a standing to a laying position, or from a walking to a running, or from an eating to a non-eating, does your blood pressure change?
Yes.
Yeah. And again, if your blood pressure goes up 5 millimeters of mercury or drops 5 millimeters of mercury, does that matter so much in your arm, in your leg, even necessarily in your brain?
No.
Probably not. But when we're talking about the glomerulus, where the net filtration pressure is 10 millimeters of mercury, is a 5 millimeter change there going to have a huge effect on the function of the kidney?
Yes.
Yeah, absolutely. It's going to have a massive effect. So what happens is we have this constantly changing blood pressure in our body, and we need to try to even it out as much as possible. And that's what happens here in our afferent arterioles. In our afferent arterioles, they have their own stretch receptors. And these stretch receptors fire action potentials to indicate how much they're stretched out. All right. And so what this allows our afferent arteriole to do is to respond to pressure changes. So let's think about this. Let's say there's an increase in pressure in the afferent arteriole. So the pressure in the afferent arteriole goes up. How is our afferent arteriole response?
Would dilate.
Well, so now I've heard both answers. It's going to constrict or is it going to dilate? Which one? What is our goal?
Decrease pressure.
To bring the blood pressure down. So to bring the blood pressure down, do I need my arteriole to constrict or do I need my arteriole to dilate?
Dilate.
Dilate, absolutely. So in this case, it will dilate. And when it dilates, that dilation lowers the blood pressure. Okay, that makes sense?
Yes.
And guess what? The exact opposite is going to happen as well. If there's a decrease pressure in the afferent arteriole, how is the afferent arteriole going to respond to that?
In this case, constrict.
And when it constricts, that increases the blood pressure. There you go. It really is just that simple. Now, you are correct. When we constrict the afferent arteriole, it lowers the blood pressure, right? It helps to lower the blood pressure, but you're right. It is also going to decrease blood flow to the glomerulus. But remember, it is decreasing the blood flow of high pressure blood. If we have high pressure blood, do we want more of that high pressure blood in the glomerulus or less of that high pressure blood in the glomerulus? We want less. So again, constricting, it responds by dilating and lowers the blood pressure. I'm sorry, I got this backwards. It increases the blood flow in the glomerulus. In this case, I'm sorry, I got sidetracked. Yeah, so that's gonna be what happens next. Increases blood pressure in the afferent arteriole. It's gonna decrease it. It's gonna lower the blood pressure. Yes, you're gonna get an increase in blood flow to the glomerulus, but of lower pressure blood. All right, so it is going to be a lower pressure blood coming in. So, and again, the goal is to maintain consistency. Here, we are increasing the blood pressure by constricting. So now we have, yes, less blood entering the glomerulus, but it's at a higher pressure. If we have higher pressure blood coming in, we want less of it coming in. So in both cases, the goal is to maintain consistency.
How it affects the filtration rate?
Well, again, filtration rate is dependent on the hydrostatic pressure. Whether you have, and again, these numbers are completely arbitrary, but if you think about it, whether you have a half a liter of blood at, 7 millimeters of mercury, or whether you have 10 liters of blood at 3.5 millimeters of mercury, right? The overall pressure is going to be the same. You want that pressure the same, whether it's more blood moving slowly against the boundaries or less blood really pouncing around on it, then you're going to get the same pressure. As long as you have the same pressure, that's all that matters. It's not the volume of blood that's in there that matters. It's the pressure of the blood that's in there that matters.
Okay.
Hold on one moment. All righty. So that is our myogenic. Questions on that? So again, remember here, the goal isn't flow. The goal is pressure. And that's our focus here. Here, the goal is to maintain consistent pressure. We'll talk about volume in just a minute, but we'll see that's what our sympathetic does for us. But here, our goal is to maintain a consistent pressure in the glomerulus, and we do that by controlling the blood pressure of the blood. So that is what our myogenic mechanism is going to do for us. Okay? Now, the second autoregulation or intrinsic mechanism is the one that we've already talked about in the last class. And that is what our juxtaglomerular apparatus does for us, right? Or what we call tubuloglomerular feedback. Because remember, what we're doing is we're doing that quality control in the distal convoluted tubule and relaying that information to the glomerulus. And what were the three types of cells that were involved in our tubuloglomerular feedback?
Macula densa.
Excellent. Macula densa cells. Remind me again what those are. Where are they found? Let's work our way through this. Where are the macula densa cells found?
In the distal tubule.
Excellent. In the distal convoluted tubule. Excellent. What are their functions? What is their job?
They have chemo and osmoreceptors.
It changed the blood flow of the
Excellent.
glomerulus.
And how do you remember how they do that? Perfect. Release nitric oxide. Excellent. So their job is to monitor the condition of the filtrate and release nitric oxide. And so again, let's see if we can remember. If it is underprocessed, does it release more nitric oxide or less?
Under, more.
And if it is overprocessed, it's going to, oops, not in, if. Release less nitric oxide. Excellent. This nitric oxide affects what cells?
Mesangial cells.
True. Mesangial cells are one of the cells that it will affect. Those mesangial cells, where are they found?
In the spaces between glomerular capillaries.
Excellent. And what do they do?
They shrink or they inflate.
How?
By actin.
There you go. They change their size with actin. Beautiful. If our filtrate is underprocessed and our macula densa cells releases its signal, what do our mesangial cells do in this instance? Are they going to build more actin or build less actin?
To expand, they need more actin. To shrink, less.
So excellent. So they're going to build actin. And when they build actin, they get larger. And that pushes up the capillary. And that increases surface area. Excellent. Whereas if it's overprocessed, they break down actin. Cells get smaller, and it's going to decrease the surface area. And what's the third type of cell?
Granular.
Granular cell, or what else we call them besides granular cell?
They're like smooth muscle cells.
True, they are, but what's their name? Granular cells, totally acceptable, but absolutely. The other name is juxtamedullary. No, I'm sorry, juxtaglomerular. Too many juxtas. Juxtaglomerular cells, absolutely. Cells, perfect. And you're right, they're the smooth muscle plus endocrine cells. And where are they found?
Afferent Arteriole.
Excellent, perfect. And when it is, filtrate is underprocessed and our maculodensa cells release more nitric oxide, what do our granular cells do? Gonna dilate, dilate, there you go. And when they dilate, we make more filtrate, right? Or more blood enters the glomerulus, which is gonna make more filtrate.
But why? If it's under process, it means that there are too much filtrate, so they need to constrict.
Oh,
right?
you're right. I'm sorry. Do we have this backwards? If they're underprocessed, we need, yeah. So I'm sorry. You're right. We have these backwards. I apologize. You are absolutely correct. So let's cheat. Let's do this one. Hold on. So dilate, which increases blood flow into that environment. So hold on, let's cheat and do this. All right, so when it's overprocessed, when it is overprocessed, release more nitric oxide. Increase the surface area so we get more filtrate. Dilates the blood vessels so we get more blood flow. We get and make more filtrate. If it's underprocessed, then we release less nitric oxide, causing our mesangial cells to break down, decrease in the actin, decrease in the surface area, and it is going to constrict, which is going to decrease blood flow. Less blood flow, less filtrate being made. So I'm sorry, yes, thank you for catching that. I apologize, I started talking about it and then got it backwards. All right. So again, we talked about this last class, we're talking about this here. I apologize for the confusion, but hopefully it makes sense now. Any questions on that? All right, excellent. So those are the intrinsic renal autoregulation processes, neural processes from those short reflexes occurring inside each individual nephron. But we still have to talk about the extrinsic neural mechanism, and we need to talk about hormones. But clearly I have not had enough caffeine, so let's go ahead and take our first break here. So we'll take our first break. Looks like it's 1:05 now, so we will restart at 1:20, and I will start the recording at that point. All right. Questions on any of that? All right, see you guys in 15 minutes. Someone asked a great question during the break, so about the lecture pictures. Again, I don't have a policy against posting those lecture pictures. I do them when I feel they're appropriate. Typically, I like to post pictures when there are, like when we're talking about the graphs, like the oxygen hemoglobin dissociation curve, or something that necessarily has a lot of anatomy to it, where it's something very descriptive that we needed to talk about that way. I typically don't. I know sometimes I write out these long descriptions and explanations and stuff, but I don't bother to post those because I feel the goal in this class is not for you to just memorize the things that I have said. It's just more to make sure that you understand these processes exercises, and most importantly, you can explain them in your own words. So again, if there's something that you feel that I'm missing or some picture, I can see if I still have it, and I'd be happy to post it. But like I said, graphs or elaborate illustrations or things along those things are things that usually I feel are worthwhile. But again, if like what we have here, where I've just written a bunch of words, I don't think that that's necessarily something that needs to be recreated onto the website, because the goal isn't to memorize my words. It's to be able to understand these things, these concepts, and say them in your own words. So I appreciate that. But like I said, if you feel that there's a picture of something that's lacking, then by all means, let me know or email me, and I will see if I still have it. All righty. So we have talked about our two, there we go, we did that. We've done our two intrinsic renal autoregulation processes. And so what that is going to bring us to is our extrinsic control. And this is with our autonomic nervous system, and specifically with our sympathetic nervous system, as you guys mentioned before. Now for this, and let's actually go back here. How do I have this arranged? So that's all one thing. So actually, you know what? I'm going to need this whole screen. So let's go ahead and get rid of that. We know we have two arterioles associated with our glomerulus. We have our afferent arteriole and we have our efferent arteriole. And we haven't talked about this yet, but the afferent arteriole is much larger than the efferent arteriole. Oops. No. Why might that be the case? What's the advantage of having a larger afferent arteriole compared to the efferent?
There's never a shortage, like of supply.
How do you mean? I like what you're saying, and I think you've got the right idea. I just can you express it a different way?
Like if there's a demand for blood in the capillary, the afferent arteriole will never like come up short because it's bigger.
Okay, excellent. So yeah, you've absolutely got the right idea, but take it from where you're going a step further. Yes, there is more blood coming in from the afferent arteriole. Okay, so there is more blood coming in because it is larger in diameter, so you have more blood coming into that. But then you have a smaller exit leaving. It almost sounds similar to what we had in the lymph nodes. Didn't our lymph nodes have more lymph coming into them than they had passageways going out? Yeah, and remember that was to increase the congestion. More congestion means more pressure, means more filtration. So you're absolutely correct. It is going to help to facilitate. It is going to help to facilitate a high pressure and a high volume of blood, because that's our goal. We want a high pressure and a high pressure, a high volume of blood in our glomerulus, so that we can have efficient glomerular filtration rate. We can make our filtrate at an appropriate rate. And this is great when we are at rest. As we know, at rest, as we've talked about, there's a large blood supply going to the glomerulus. So we have a high flow of blood, high pressure blood, and we maintain an appropriate rate of filtration. Perfect, excellent, spectacular. But here's the problem, as we've talked about before. What happens when we start to exercise? When we have moderate activity, As we know, blood gets redirected to other parts of the body. And so what that means is we have a much smaller volume of blood that is going to be entering into our glomerulus. So when this occurs, remember this is when our sympathetic nervous system is active. And when our sympathetic nervous system is active, what our sympathetic nervous system does is it leads to a constriction, both the afferent and the efferent arterioles. So let's think about the effect on that. Here, when we constrict, what is that effect going to have on the blood coming in?
Flow decrease.
I'm sorry?
The flow decreases.
But see, remember, the flow is already decreasing. We already have less blood coming in. So when we constrict the blood vessel, we're not necessarily getting less blood to come in, but what are we doing to the blood that is coming in? Alexa's got it. When we constrict here, we are going to increase the pressure on the blood that remains. Yes, potentially that could reduce the flow, but remember the flow is already being reduced. We have less blood coming in here. So when we're constricting it, we're not limiting the blood that can come in, but we are doing is we are increasing the pressure of the blood, of the, and let's be more specific, of the reduced volume of blood that enters the glomerulus. Excellent. And what effect is constricting the efferent arteriole going to do?
It's about flow and pressure, right?
True, although in this case necessarily is it so much going to be the pressure in the afferent arteriole that we're concerned about? When we constrict our afferent arteriole, what is this going to affect?
The flow?
Yeah. Here when we constrict, this is going to reduce the flow of blood out of the glomerulus. Bingo, you got it, Nick. When we're trying to reduce the flow of blood out of the glomerulus, this means the blood is in the glomerulus for a longer period of time. And remember, if it's bouncing around in there, that increases the turbulence of the blood, which, as we know, increases the pressure. So as you guys have pointed out, what happens here with these two constrictions of both the afferent and the efferent arteriole, what happens is we have much less blood coming to the glomerulus, but we still maintain the high pressure needed for proper filtration. So in that active situation, our sympathetic nervous system allows us to constrict both the afferent and the efferent arteriole. And that is going to help us to maintain a higher pressure, even though we have much less blood coming to the kidney is still going to be able to function normally, even when you're riding your bike, even when you're walking to the store, even when you're walking your dogs, and everything else like that as well. All right. Questions on that? I have a question. Notice I said this works under moderate activity. What about extreme activity? What happens in that situation? In extreme activity, when you're fighting that bear with an axe, when you're sprinting, with that extreme activity, when you have an extreme decrease in blood going to the kidneys, is this still going to work? Are we going to be able to constrict those arterioles enough to be able to make up for that? No, Nick's got it, absolutely. There's only so much we're going to be able to squeeze them. So in those extreme situations with extreme activity, there is a decrease in kidney function. Now, that is a bad thing, but at the same time, how long are you sprinting for? Anybody here able to sprint for 4 hours or more? An hour, 10 seconds sounds about right. Absolutely. If you're fighting that bear with an axe, right, how long is that going to last for? Right, not very long. So luckily, most extreme stressful situations aren't long lasting. However, right, again, stress used to be related to things that threatened our life. If you have some type of very high stress job or class or something along those lines, can chronic high stress have an impact on our overall kidney function in time?
Yes.
Yeah, absolutely. So chronic stress is something that does have to be a concern for individuals with their kidneys. If you're in a very high stress job or a high stress situation, that chronic high stress is something that could potentially have long-term impacts on your kidneys. But typically, the type of extreme stress that is directing blood to other parts of the body to deal with those extreme physical exertions don't last for a very long period of time. And so they're typically not going to have the same, while they will impair kidney function. If you're fighting a bear with an axe, making a little less urine is probably the least of your concerns. All righty. Questions on that? All right. And remember, it doesn't matter how fast the bear is, Nick. You don't have to outrun the bear. You just have to outrun one other person. All right. Excellent. All righty. Now, when we're getting that afferent arteriole to contract with the autonomic nervous system, the other thing that it does is it stimulates those juxtaglomerular cells, the granular cells, to produce our hormones. And the hormones are the third regulatory mechanism. Now, the good news is some of these things we already know a bit about, although we can improve our understanding of them, starting first with our anti-diuretic hormone. I'm going to cheat and hide all of this with a box so we can do a little writing. What do we know anti-diuretic hormone does?
Lower blood pressure.
True, absolutely. It lowers blood pressure.
And blood volume, right?
How? Well, is it lower blood pressure and volume or raise it? I mean, you were correct. Let's say it this way. It affects blood pressure and volume, right? Let's say that it affects blood pressure volume. If you increase the amount of antidiuretic hormone, then you increase blood volume, increase blood pressure. If you lower the amount of antidiuretic hormone, then it's going to do the opposite. It's going to decrease blood volume and decrease blood pressure. Because what is it that antidiuretic hormone actually does?
It's about sodium, that water follows sodium.
Actually, antidiuretic hormone is really more about water. It's not sodium. In this case, it is about water. What antidiuretic hormone does is it tells the kidneys to reabsorb more water, right? That was what we said before. When we were learning about it in the endocrine system, we were mentioning how it tells the kidneys to absorb more water, right? That's how when we were talking about when you drink that caffeine, when you drink that alcohol, that suppresses the release of antidiuretic hormone. And remind me again, where is antidiuretic hormone made? Antidiuretic hormone. Where is antidiuretic hormone made?
In the kidney. No?
The brain? Antidiuretic hormone? Can we be a little more specific than the brain? Was it made in the anterior pituitary? Was it an anterior pituitary, posterior pituitary?
It's the anterior.
Let's see. I've seen kidneys. I've seen anterior pituitary, posterior pituitary, brain. None of them are the right answer, though. Where is antidiuretic hormone made? There we go. It is made in the hypothalamus. Where is it released from?
Anterior.
Nope, posterior. Posterior pituitary. Excellent. Perfect. We said its target was the kidney, and it told the kidney to reabsorb more water. But now we can be more specific about how it does that. Where it actually targets is in the distal convoluted tubule. In the distal convoluted tubule, basically what it tells the distal convoluted tubule cells to do is to absorb more water. The distal convoluted tubule, for the most part, is impermeable to water. Normally, we talk about how water can just easily pass through the phospholipid bilayer, but not in the distal convoluted tubule, not in the collecting duct. For the most part, those areas are impermeable to water. So what antidiuretic hormone does is it tells those cells to express a special channel on their apical surface. And that special channel is called an aquaporin. Cells of the distal convoluted tubule, cells of the collecting duct. It tells them to express an aquaporin. It's a special channel, and guess what can travel through the special channel that is an aquaporin? Water, exactly. And this allows water to be reabsorbed. Remember, we mentioned how the distal convoluted tubule is where our fine tuning is going to occur. That fine tuning is primarily going to occur driven by hormones. So we're going to get selective processing, hormone-driven processing that is going to occur. And antidiuretic hormone is one of the examples of that. It causes the expression of those aquaporins so that water can be reabsorbed. And when we reabsorb water, It increases our blood volume, it increases our blood pressure, and it not constricts, it concentrates our urine. Now, questions on that? Not too much new information there other than the aquaporins. Although I'm sure after reading the exams, most of you were hoping that you wouldn't have to deal with hormones again, but lo and behold, here we are. All right. Then let's talk about the next one, atrial natriuretic peptide. Where is that produced again?
In the atria.
And what does it do?
It lowers water and sodium in blood.
Right. And so the way it does that is it tells the kidneys not to reabsorb water. So what atrial natriuretic peptide does is it actually blocks the expression of those aquaporins.
So volume decrease and blood pressure decreases, right?
Yeah, you block the expression of the aquaporins, less water is reabsorbed. That is going to lower the blood volume, which of course lowers the blood pressure. And again, indirect effect is it is going to give us a larger, more dilute urine. Essentially, it's a diuretic. An anti-diuretic is an anti-diuretic. Atrial natriuretic peptide is a diuretic. One has us hold on to water, one has us release water. Again, with the exception of the aquaporins, again, not a lot of new information here. The one we've mentioned a couple times and haven't talked a lot in depth about yet is this renin angiotensin aldosterone system. So here's where we have to talk more about this process and get more in depth in what's going on. Not surprising, the trigger for this is going to be, oh, we did all of this. Anti-diuretic hormone, water absorption in the principal cells of the collecting duct, distal convoluted tubule. We did that. We did that. And atrial natriuretic peptide, I should also mention, inhibits reabsorption of water, stopping that water loss, that water reabsorption. All right, so we had this here, but we did that already. All right, this is the one I want to talk about. Renin angiotensin aldosterone system. In the case here, the trigger for this is typically a drop in blood pressure or a drop in blood volume. Right. This, of course, occurs in the kidney, and more specifically, this occurs in the afferent arteriole. When we have a drop in blood pressure, when we have a drop of blood volume, as we know that those afferent arteriole, those granular cells are going to constrict. And when they constrict, they are going to release renin. Remember, as I mentioned, renin is not a hormone. It is a protein, but it is a protein that acts very much like a hormone. So it is a hormone-like protein. One of the main effects of that is it activates a protein called angiotensin 1. Angiotensin 1 is produced in the liver. However, it is converted into angiotensin 2 in the lungs. So angiotensin 1 is the inactive form. Angiotensin 2 is the active form. And what renin does is renin turns on or activates, It activates the enzymes, well, that's wrong, in the lungs that converts angiotensin from its inactive form, angiotensin 1, into its active form, angiotensin 2. So
What
again,
enzymes- I'm sorry, what enzymes of the lungs activated?
there are several of them, and you don't need to know the names of them.
Okay.
But just know that renin activates or turn on the enzymes that convert angiotensin 1 into angiotensin 2. Okay? Now, the question then becomes, what is it that angiotensin 2 does? And it turns out it does a lot. No, great question, Jessica. So it doesn't activate the angiotensin 1. Angiotensin 1 is already there. What renin does is it activates the enzymes that convert angiotensin 1 into angiotensin 2. So 4 steps. So let's see. Okay. Drop in pressure in the kidney, releases renin. Renin activates an enzyme, and that enzyme converts angiotensin 1 into angiotensin 2. Sure, I like that. 4 steps. Now again, you don't have to do that in 4 steps. You can discard that however, but that works for me. All right. But let's get to the fun part. What angiotensin 2 does. Many effects, and we'll see the influence and overall effect of all of them. One of the things that it does is it constricts systemic arterioles. If we constrict systemic arterioles throughout the body, let's think over here in terms of what the effect of all of this is going to be. If you constrict systemic arterioles, what is the effect of that going to be?
Increasing in blood pressure.
Excellent. Increases blood pressure. Perfect. It tells the kidneys, angiotensin 2 tells the kidneys, again, primarily in the distal convoluted tubule, where things are controlled hormonally, to absorb more sodium, more chloride, and of course, where the ions go, water follows. So what would the effect of that be?
Volume drops.
Okay, right, it's going to increase blood volume. And of course, if we increase blood volume, then that is also going to increase blood pressure. Excellent. It triggers, where does aldosterone come from again? Adrenal gland, and you even got adrenal cortex there. However, we can probably be a little bit more specific. What specific region of the adrenal cortex? I'll give you a hint. It starts with zona. Granulosum? Close. It is a G.
Glomerulosa.
There you go. Excellent. To release what is a class of hormones. What was that class of hormones that aldosterone is a part of?
Androgens.
No, it wasn't the androgens. What was the aldosterone is the specific type of a class known as
Somatocorticoids.
well, okay, sure, absolutely. The corticoids would be a more general term, but I was looking for mineral corticoids. Excellent. That class of mineral corticoids, aldosterone, and aldosterone tells the kidney, targets the kidney, and again, targets the distal convoluted tubule to do what? Reabsorb sodium, absolutely. And then, of course, wherever that sodium goes, what's going to follow?
Water.
Excellent. And of course, once again, if we've got water coming back, we are going to increase blood volume and blood pressure. Should be sensing a theme by this point. Angiotensin 2 also stimulates your hypothalamus, specifically an area that makes you thirsty. And typically when you're thirsty, what do? You drink water, absolutely. And when you drink, that increases more water into your blood. So that is going to increase blood volume and therefore blood pressure. Excellent. So someone's asked, is this the dehydration trigger? Yeah, so I'm not, I haven't heard the phrase dehydration trigger, but yes, this thirst center in your hypothalamus, when active, does give you the sensation that you are thirsty and that you want to drink. Absolutely. So it does do that. I'm not sure. Again, dehydration trigger may be a term. I'm not familiar with it, but that doesn't mean that it isn't a term. But yes, this thirst center helps to regulate your desire to ingest fluids. And notice this is also going to stimulate the release of antidiuretic hormone. And we all already know antidiuretic hormone is going to increase water retention. So notice once again, that is going to increase blood volume and therefore indirectly increase blood pressure. So notice we have direct effects on blood pressure, and then all these effects on blood volume, which of course indirectly affect blood pressure as well. So all of these effects of angiotensin, all being driven by that drop in blood pressure, that drop in blood volume, helps to bring all of that up. And again, like we see with hormones, we get a big global effect. Now, I like my list here, but your book has this really, really cool looking flow chart that shows the same thing. Right here we have that decrease in blood flow to the kidneys, right? That drop in filtration rate, urine production, and our juxtaglomerular cells, those granular cells, release renin. All right. It constricts the afferent and efferent arterioles, or efferent one. So again, increase in glomerular pressure, increase in glomerular rate. If it tells the adrenal gland to release aldosterone, increase sodium retention. Thirst center, so we drink more, more antidiuretic hormone, right? We get our vasoconstriction, increase heart output. All of these things increase our blood pressure, increase our glomerular pressure, increase our blood volume, so that our glomerular filtration rate comes back. Great question. So if you were treating high blood pressure by taking a high blood pressure medicine, would it have an effect on this pathway? Yes, but again, I think you've hit on the key there. If you have high blood pressure, if you have chronic high blood pressure, we often talk about the problems that that chronic high blood pressure has on our heart, making our heart have to work harder. But if the glomerular pressure is up as well, if that hydrostatic pressure in the glomerulus is 65 instead of 55, then that means that's going to be putting a lot of strain and a lot of excess work on your kidneys as well. So yes, chronic high blood pressure can actually cause a condition called nephritis, where we get an inflammation of our nephrons. And when they become inflamed, they become more permeable, more larger molecules can get out. That can be one of the things where we see large formed elements getting out, bigger proteins and things like that getting out, and that can cause problems as well. right? If you have high blood pressure and you have more stuff being forced out, then you're going to be making filtrate at a much, much too high rate. You're going to be under processing it. You can, like I said, it could be damaging the glomeruli. So there's all those problems associated with it. So taking that blood pressure medicine to compensate, to bring that blood pressure down would be beneficial. Now, if you're taking blood pressure medicine for the high that it gives you, and I don't know if it gives you high, but I'm just saying, and you are artificially lowering it, then that would be a problem as well. But usually, I'm pretty sure there isn't a kick associated with blood pressure medicine. So typically people are taking it to bring that blood pressure down. If there is a kick, let me know, because I think we still have some of my grandfather's old pills. I'll start taking those. Anything to pass the time while I'm here at home. All right. I'm just kidding. I don't need to do that. Marijuana is legal now. All right. Questions on that?
I want to ask.
Yes.
So is there a short way to describe it, or you want us to describe it, everything that's mentioned here on the table?
Sure. The short way to describe it is that it increases blood volume and blood pressure. However, if I ask you in an essay question to tell me what the renin, renin angiotensin aldosterone system is, and you tell me that it increases blood pressure, is that going to get you all the points on the exam?
No.
No. So give me the details. I want the details. All right. Questions on that? All right, excellent. And again, and more, I love this picture too. This is a great illustration from your textbook, putting it all together. Notice here we have the intrinsic mechanisms where we have that myogenic control, that tubular glomerular, so intrinsic neural. Here we have the extrinsic neural, and here we have the hormonal. Do a great job of showing how all these things respond to, for instance, a drop in blood pressure. So it's a great, great picture from your textbook. All righty. Questions on that? Sorry, my daughter is doing her Zooming for school outside, and there was a caterpillar on the chair near her. And so she wanted to know what it was. And so I was just explaining to her that it was poisonous and that it can jump 10 feet in the air to try to get to her neck. So she needs to be careful. All righty. Excellent. All right. So with that, we have learned everything we need to know and more. No, just a little caterpillar. Everything that we need to know and more. Here, she sent me a picture. You see that okay? Yeah. So you can tell it's got the red bit on it, so it's totally deadly and poisonous. All right. Exactly like yes. Run away. All right, excellent. For some reason, she doesn't believe me. All right, excellent. So we now know everything you need to know and more about glomerular filtration. We know how to make our filtrate. So now we need to focus on the two processes of tubular reabsorption and tubular secretion. And the good news is, for the most part, the two mechanisms are very similar, right? They're both active primarily. They both are moving substances. They're both selective. The main difference between them is they're going to go in different directions. So obviously the first main question is, what type of substances are we talking about? Notice for reabsorption, we have all of those big important metabolites, things like glucose, things like amino acids, things like proteins, things like vitamins. Those are all things we need. And then notice the other big thing that we're both reabsorbing and secreting are ions, because remember, the composition of our blood plasma is the composition of our interstitial fluid. So notice we need to make sure we have nice high levels of sodium. We need to make sure that we have nice high levels of chloride and magnesium and bicarbonate and sulfates and all those kind of things as well. However, notice when we look at our lists here, both potassium and calcium in particular are on both lists. Because as we know, potassium, we want to be at low levels. So while we need to bring some of it back, we don't want too much back. We want to keep it relatively low in the interstitial fluid. And calcium, as we know, we have to, make cells do wonky things. Again, this was not one of the numbers that I made you memorize way back in 430. But if some of you may remember in 430, I did mention it briefly. Again, this is not a number you need to worry about. But remember, the range of calcium, the appropriate range of calcium in our blood is between 11.5 and 8.5 milligrams per deciliter in the blood. And again, that isn't a number that you have to memorize. The only reason it sticks in my mind is because of how narrow a range that is. And so again, it's not about having all the calcium or none of the calcium. It's about having precisely the right amount of calcium in our blood. And so not surprisingly, those are things that are going to be both very actively reabsorbed and also very actively secreted as well. Also, obviously, we want to get rid of wastes, biological wastes, organic acids and base, ammonias, creatine, other things like that, that we are producing from that metabolism, as well as a bunch of random other things. Those medicinal and recreational drugs we were just talking about. But notice also our neurotransmitters, our hormones, right? Many hormones are going to be too large to fit through, but some of the smaller amino acid derived hormones can be released by the kidneys. Remember, anything that's not or anything that's too big will be broken down by the liver. But also neurotransmitters that get released, histamines, things along those lines can also be released as well. So again, this is not an all-inclusive list, but it gives a good indication of the types of things that we are talking about. And like I said, we're putting 200 liters of filtrate into that nephron, into that kidney. And so we need to reabsorb around 99% of that. And as we keep talking about it, about 60 to 70% of that reabsorption is going to occur in the proximal convoluted tubule. Now, when we talk about this process into the filtrate, again, we need to move the fluids between the blood and the vessels, the blood vessels and the tubules. And there are numerous methods that we are going to use for this. Some of it will be passive transport. Filtration obviously is a passive process like we talked about, but we can also take advantage of simple diffusion for things like gases, facilitated diffusion where things like glucose or sodium or things like that can be reabsorbed, and even osmosis for the movement of water. As we know, things move down their concentration gradients. And when we talk about water, water moves based on its osmolarity. And remember, osmolarity is the measure of the amount of stuff. That is measured in milliosmoles per liter. And as we'll see, in normal blood, normal blood has an osmolarity of about 300 milliosmoles. So normal blood, interstitial fluid, has about 300 milliosmoles. And that is a number we are going to need to know. Of course, we're not going to rely just on passive transport. There's going to be a whole lot of active transport as well, including our pumps, our exchangers, our co-transporters, and our antiporters. Again, these are not new mechanisms. These are things that we have talked about when we were talking about, nervous system back in 430, when we were talking about digestion, when we were talking about, the action potentials in the cardiac muscle, all of these things. These are not new mechanisms that you are not familiar with. Now, as I keep saying, primarily, it is going to be driven either directly with primary active transporters or indirectly for our secondary active transporters by ATP. And in fact, when your body is at rest, like you would sitting commonly here in the classroom, half asleep from listening to me drone on, somewhere around 20 to 25% of your body's oxygen is being used by your kidney to filter that blood, to produce that ATP to filter that blood. So again, which is not surprising because you got about 25% of your blood going to the kidneys, carrying 25% of the oxygen. That seems reasonable. Now, as we see nicely from this illustration, there are going to be a couple of paths that can be taken for our resources to be reabsorbed. One of the things that we have here, let's actually go back a second. There we go. What we have here is a picture illustrating what is here, of course, the lumen, and that lumen contains the filtrate. And much in the same way when we talked about digestion, there are basically three membranes that must be passed to reabsorb a substance. Those three membranes are, of course, the apical surface, the basal surface, and then, of course, the third one is the endothelium of the blood vessel. Right, because we know it needs to get back into that peritubular capillary. Let's say that. The peritubular capillary. Excellent. So those are the three things that it has to pass between. Now, much like we saw in the digestive system, there are tight junctions. Remember, the tight junctions are these big, huge, thick proteins where we actually have multiple proteins. Let's actually undo this. Transmembrane proteins that from one cell connect to transmembrane proteins of the other cell, forming a zipper or mesh-like connection that anchors the two cells together. And as we've talked about, the advantage of this is twofold. One of the things that these tight junctions are going to do is they're going to limit, notice they don't stop it entirely, but they limit the amount of stuff that can pass between the cells. But the other thing that it does is it also locks the phospholipid bilayers. Remember, as we've talked about, our phospholipid bilayers are a fluid mosaic. They're constantly switching places with each other and moving things around. And as they move around, the proteins and the carbohydrates and the things like that that are embedded within them get moved around as well. But by putting this boundary on them, what it does is it maintains the different channels and transporters on the two sides. So basically what's on the apical side stays on the apical side. What's on the basal side will stay on the basal side. So a sodium glucose co-transporter on the apical surface isn't suddenly going to move to the basal side. It keeps the channels and the transporters separate. So what's on the apical side stays on the apical side. What's on the basal side stays on the basal side. All right. But again, the key word there is limits. There indeed are still going to be some things. Here we see sodium sneaking its way in between. And when it sneaks in between the cells, that is our paracellular reabsorption, going between the cells. For some ions, as much as 50% of that ion can be absorbed that way. Potassium is a good example. About 50% of potassium can be absorbed this way by sneaking in through the cracks. Chloride can do that as well. So some ions are able to sneak in paracellularly. But many substances need to be transcellular in their reabsorption. Again, they have to pass through the apical layer, they have to pass out the basal layer, and typically the mechanism for both of those are going to be separate. And then lastly, it has to enter into the endothelium of the capillary. All right, because again, once we get here, we're just in the interstitial fluid. Up here, they've written it. Just in the interstitial fluid. So once you're in the interstitial fluid, if you want to get back into the blood, then you have to get into the capillary. But as we'll see, not everything that gets back into the interstitial fluid wants to get back into the blood. So we'll see how that works as well. All right. Questions on that?
So paracellular is passive and transcellular is active?
Great question. Paracellular absolutely is passive. Right, we're not going to be using any ATP for it to go through that way. Transcellular can be passive or active. Right? If for instance a water molecule passes through here, that would just be simple diffusion. That would be passive. If oxygen just passed through there, that would be simple diffusion, right? However, if there was, like a sodium channel, then it could just pass through that as well. So this could be passive, but it's also, there's going to be those active transporters, those pumps, those things that way. So yes, with transcellular, it can be passive or active, but you're absolutely right. Paracellular is always passive. And we'll actually talk about specific examples. We will actually see these in action. We're just kind of setting the table here so that we can see the processes that are going to be involved. All right. Now, when we talk about a carrier-mediated transport, again, what we're talking about, these are things like pumps, these are things like channels, these are things like co-transporters. There are limits to how many things can travel on them and how quickly. It's like when you're at the mall, right? Back in ancient time when the malls used to be super, super crowded, right? If you were at the mall, downstairs at the mall, and suddenly there was an announcement that One Direction was finally getting back together and they were doing a free concert up on the second floor of the mall, then you would have a massive humanity trying to get from the downstairs to the upstairs at the mall. And of course, there's two ways you could do that. There is the stairs, where you have to use your energy to get up there, right? That would be a passive transport because the mall is not wasting any energy. But there's also the escalators and the elevators. Those are things where the mall is providing the energy and you get to just stand there and ride. But Even on that elevator, even on that escalator, even on those stairs, are there limits to how many people you can have going up them at once? These are the easy questions. Absolutely, yes, absolutely there are. And that limit is what we call a transport maximum. Every single protein, whether it is passive or active. So this is true for both passive and active transport. There is a limit on how many can go. Obviously, we're just talking about moving in one direction. Everybody wants to get upstairs to see one direction. And not surprisingly, what's on the apical side is different from what's going to be on the basal side. So typically, when we're talking about transport maximum, just like in digestion, where it matters is on the apical side. Because if you don't get in the apical side of your renal tubule, what happens to you? If you stay in the lumen of the renal tubule, what happens to you?
You become urine.
Yeah, you leave the body with your urine. We don't get it. Once we get it across the apical side, we've stopped it from leaving in the urine. And so just like in digestion, where we're going to use more of our energy on the apical side, we usually don't have to use it on the basal side, because on the basal side, we've already kept it. We've stopped it from being lost. So that upper limit on the apical side is what is known as the transport maximum. Transport maximum is typically measured in milligrams per deciliter. And every single substrate has a transport maximum. For glucose, for instance, it is 180 milligrams per deciliter. For amino acids, it's 65. What's nice about this is that very easily then, if you know the concentration of a substance in the blood, you know if it should be in the urine. Let's take glucose for an example. Anybody here know what a normal concentration of glucose in your blood should be? What is a typical healthy, good glucose level? Come on, I guarantee even in a class this small, someone either has or knows someone who is monitoring their blood glucose levels. Excellent. 100, 80, 7, 100, I like that. So if our transport maximum for glucose, if our filtrate is able to move out 180 milligrams per deciliter of glucose out of the filtrate and back into the body, and the average person has 100 milligrams per deciliter of glucose in their body, should there be any glucose in their urine? No, because we're able to draw it all out, and so there should be zero of it in the urine. But what happens if someone isn't able to control their glucose levels? Like, for instance, with diabetes mellitus. In those instances, somebody's glucose level can get as high as 400 milligrams per deciliter. What happens when that glucose level gets up in the 400 range? Is there going to be glucose in the urine then? Absolutely. In fact, while we haven't understood the concept of transport maximum, this is something that we've understood since medieval times. Medieval times, if you had a baby that was really upset and really crying, one of the things that the doctor would do was taste the urine. All right, that's why doctors make so much money. They're the ones that have to taste the urine. And of course, what was he tasting that baby's urine for? Tasting it for the sugar, seeing if it was sweet. That would be to see if it could find out, right, that if there was glucose in there. All right. So by knowing the transport mechanism of a solute, and to get his teeth white, exactly. So if we know the concentration of a solute in our blood, and we know the transport maximum, we can know whether or not it should be in the urine or not. All right. So again, we talk about glucose, and we'll actually talk more in detail about the pathway for this here. But as you can see, glucose is brought in by a co-transporter on the apical surface with sodium. Our friend sodium is always going to be the one that brings it in. Notice we then have to pump that sodium out so we maintain that concentration gradient. So sodium wants to continue to come in. And then once the glucose is in, it can just leave via facilitated diffusion. And in a healthy individual, we're able to reabsorb all of that glucose. 100% of it's going to be reabsorbed. But in a condition like diabetes mellitus, you're going to have glucose in the blood. And of course, we call that condition glucosuria. I have a cool animation I stole from another instructor that I like that shows this. Work. Why are you not working? Interesting. Oh, I've never showed it this way before. All right, so here we have the case of someone who has glucosuria, someone who has uncontrolled diabetes. In this case, what happens is our transport maximum is reached, and not all of the glucose is going to be able to be reabsorbed. Notice there's another key factor with that. If more glucose stays in the filtrate, what's going to happen then? If we have more stuff in the filtrate, what's going to follow that stuff into the filtrate? Water, absolutely. Oops. Water. Oops. There we go. Water is going to be drawn to that as well. So typically someone who has uncontrolled diabetes typically has not just sweet urine, but typically have a larger volume of more dilute urine as well, which can lead to dehydration and all sorts of other issues as well. There you go. Polyuria. All right. Questions on that? All right. Let me do something real fast here. All righty. So honestly, that was everything that I wanted to cover for today. We do have a little bit more that we could do. It's a lot more though. So I think what we'll do is get a little bit ahead so we have a little less to do on Tuesday. I don't think we have a full hour's worth of material left, but again, the less we have to do 2 days before the exam, the better. So I do want to go through this last concept. So I think the best choice of action right now is to go ahead and take one last break. Take a 15-minute break. It's 2:25 right now. We'll come back at 2:40. And at 2:40, we will start the process of tubular secretion and start talking about some of the specifics. And then we'll save the rest of it for Tuesday. But I think the best course of action is for us to continue forward with this. So, oops, what just happened? So I think that'll be a good game plan. So I think that's going to be the smartest way to do this. So let's go ahead and take a break, come back at 2:40, and 2:40, we will talk about the precise mechanisms for which it is. the 3 dodges, or 4 dodges, dodge, duck, dive, dodge. What did Patches O'Hoolan say? I don't know how that relates to urine. All right. So short break, come back, and we'll finish the rest. Any other questions? Awesome. Don't do that. We'll talk about why that's not important. That's a good not to do when we talk about actual what urine is. All right. Any other questions? Excellent. So I will see you guys back here at 2:40. All righty. So like I said, this one shouldn't take us too long, but I wanted to come to it fresh. So I wanted to make sure that we have it. There are basically 5 main processes that are going to occur in our proximal convoluted tubule. Remember, that's where most of our processing takes place. And all that processing can pretty much fall into one of 5 categories. Now, the first of these is reabsorption of organic nutrients. So again, in this case, and we can cheat and come back to my drawing here. Here we have my drawing. This is the wall of my proximal convoluted tubule. So as you know, it's got a large number of mitochondria in them. It has an apical and basal surface that are separated by our tight junction. Out here would be our lumen, which is where our filtrate is located. And then back here is where the interstitial fluid and the blood vessel would be. Now, there are going to be some specific pathways that you will need to know, just like we talked about specific pathways in the digestive system. There are going to be some specific pathways here, but those we will save for Tuesday. So right now, I just want to talk about the general classifications. And the first major process that is occurring is the reabsorption of organic nutrients. This is things like amino acids. This is things like glucose. These are things like other types of metabolites like that. These organic nutrients are primarily going to be active. So we're going to be using things like pumps and co-transporters. So for instance, if it was something like a glucose, as we know, that would use one of those co-transporters where we would let sodium come in and it would bring in a glucose with it. But they could also be things like we'll do it as a square, a glucose facilitated diffusion. As well. So this is primarily active, and for the most part, 100% of our organic nutrients are reabsorbed in the proximal convoluted tubule. But remember, as we also talked about, we also need to have that reabsorption of ions. Maintaining the ion flow is the appropriate ion concentration of our interstitial fluid and of our blood is going to be very, very important, especially sodium, but also chloride, right? As we talked about, less so for things like potassium and calcium. But once again, we are going to have active pumping, and as well as, like I said, the facilitated diffusion. And those co-transporters. After all, if we're using sodium to come in with glucose, then we're letting sodium in as well. So this works for our sodium as well. Oops, hold on. Blue. Wrong blue. Oh, this shouldn't have been blue. This should have been green. Something like that. That would have been clever. Oh, I'm not clever. Oh, so we have that. We have, again, facilitated transporters, pumps, things along those lines. Of those things coming in. And notice these are both transcellular. Now, with this transcellular movement, and now I will cheat and use a lighter color blue, there is a lot of stuff being moved out of the filtrate back into the cell and back into the interstitial fluid. And as we know, when there is a lot of stuff moving in one direction, what likes to follow stuff?
Water.
Water, absolutely. So what we're going to get here is our reabsorption of water. It is following all that stuff that is actively being moved back. It's absorbed. Now, if salt and glucose and amino acids and all those things are going back, water has to follow it. It doesn't have a choice. Wherever the stuff go, water needs to follow, right? And so this type of movement of the water is what we actually call obligatory water reabsorption. What does obligatory mean?
It's obligated to go.
It has to go, right? It has to go. Obligated means that it has to go, right? Thanksgiving, thankfully for COVID happened. Again, I can't believe I just said that out loud, but, right, Thanksgiving is right around the corner. And so every year, my brother-in-law likes to do a big Thanksgiving at his house where they invite all of their family and all those kind of things. And no room for my family, but plenty of his family and kids and cousins and uncles and all sorts of things like that. And do I have a choice as to whether or not I want to go? No, I am obliged to go. Absolutely not. I don't have a choice in the matter. I must go, right? It is obligatory. And that's what's happening here. Water doesn't have a choice in the matter. That water is being forced to follow all of that stuff. So we get this obligatory water reabsorption where it must follow the stuff. It has no choice in the matter. Now, this reabsorption of water does something interesting. If you think about it, with all, oops, this doesn't need to be all in caps. With lots of water leaving the filtrate, what ends up happening is it concentrates the remaining solutes, right? If you have a bucket full of salt water and half of the water evaporates and leaves, what does that do to the saltiness of the water that remains? Gets even more salty. It concentrates the remaining solutes, and that produces a large concentration gradient. And concentration gradients, as we know, are very good for driving passive diffusion. And so that's what's actually going to happen. The first thing that's going to happen here is we are going to get that passive paracellular reabsorption, thanks to this increase in the concentration of the remaining solutes. we're going to be producing a concentration gradient, and this concentration gradient is going to help to drive things like potassium, things like, magnesium, hydrogen ions, things along those lines, urea, these types of things. And again, the problem with this, because this is passive, some of this stuff can be good stuff and some of this stuff can be bad stuff. And this is also why we need to then later actively pump some of that stuff back out. So we are going to get that passive paracellular reabsorption that is going to sneak through between the cracks. And then, of course, what is our fifth process that needs to take place here in the proximal convoluted tubule? I kind of just hinted at it. There you go. Tubular secretion. So tubular secretion, right, which is, again, of course, obviously the movement of things that we don't want, things like urea that sneak themselves in, or other larger waste materials, toxins, drugs, things along those lines are going to be kicked out of the cell. And again, this to be selective is typically going to use some type of ATP, either directly or indirectly, to be able to move those things out. So again, I've done it here with the pretty picture, and this picture I will say for you. But let's go back to the lecture and talk about this as well. Oops, share that, thank you. So like I said, the 5 major processes that are occurring in our proximal convoluted tubule is first the reabsorption of organic nutrients, things like glucose, things like amino acids, right? Other types of metabolites. Again, primarily using co-transporters actively, or some facilitated transport as well. The active reabsorption of ions. With the movement of ions, this is primarily going to use either primary or secondary active transport to get these things where we want to go, using a large amount of ATP like we talked about. And again, the movement of these metabolites, the movement of these ions is a whole lot of stuff going back into the cell. And that is going to be a huge driving point for, oops, there we go. To get regulated by ion levels of the blood hormones, blah, blah, blah. Reabsorption of water. Again, through aquaporins. However, one of the keys to these are static aquaporins, meaning that these are independent of antidiuretic hormone. These are present all the time. So here in the proximal convoluted tubule, oops, these aquaporins are going to be present all the time. Whereas remember, in our distal convoluted tubule, antidiuretic can cause them to be expressed or can cause them to be pulled back. Again, this reabsorption of water is the water being forced to follow the salts, water following all that stuff. We call it that obligatory water reabsorption. And notice 65% of the reabsorption of our water. So of those 200 liters that are reabsorbed, that are expressed into our filtrate, 130 of those liters go back right here in the proximal convoluted tubule, following all this stuff. It has no choice in the matter. There's no question about whether it does it or not. It must be reabsorbed this way. It must follow the stuff. Now, the effect of that is that it increases the concentration of the remaining solutes, setting up a nice steep concentration gradient for us. And we can use that concentration gradient to allow for that passive reabsorption of the ions, primarily through our paracellular cracks between the cells, but there can be some leak channels that it passes through as well. And then lastly, and again, not leastly, we will talk about it, but we will save that for Tuesday as well, the process of secretion, which is, like I said, basically reabsorption in reverse, the movement of materials out. All right, questions on that? All right, with that, that is everything that I wanted to cover today from a lecture standpoint. We only have a little bit of a lecture left. We are going to talk about specific pathways of materials that you are going to be responsible for. We'll talk about secretion. We will then talk about what happens in our distal convoluted tubule. We'll talk about what happens in the loop of Henle with our dilution or concentration of our urine. And then all we have left is the storage and transport of it through our urinary tract and micturition. And guess what? Your micturition reflex, you already know. It's going to be basically the exact same mechanism that we use for defecation, is essentially the same type of mechanism we're going to use for your micturition, the voiding of urine from our body. All right, questions on any of that? So we only have a little bit more to cover. Obviously, this is not going to take us the whole class time on Tuesday, which A, is a good thing because it's 2 days before the exam, but B, that also means that'd be a great opportunity for a question and answer review. So any questions you have on the respiratory system or the urinary system, have those prepared for Tuesday to help you to prepare for the exam on Thursday. Notice we've got about 3 lectures of respiratory and 3 lectures of urinary. So I would say it's probably going to be an even 50-50 split between the 2 topics for this exam, both on the lab exam and on the lecture exam and the essay questions. I would say probably a little bit more histology for the urinary system than for the respiratory system. Not a huge amount for either, but probably a little bit more for the urinary than for the respiratory. So there's going to be a fair amount of that. But there's probably more gross anatomy for the respiratory than there is for the urinary. So be thinking about those things over this weekend. Study hard, study well. If you have questions, as always, come to my office hours or send me emails and I will respond to those. All right. Any questions?
Will you give us more time for this exam? I mean, lecture exam.
Yeah, well, so again, the amount of time for the lecture exam is totally dependent on the number of questions that we have. That last exam was just on 1 section. There was only 25 multiple choice questions. There was only 7 essay questions. So it was fewer questions, so it was less time. If this one has more questions, this one will have more time. So there isn't a set time. It's the same thing for the lab exams. Neither the lab exams nor the lecture exams have a set amount of time. They have to fit within our classroom time. And I do like to have students allow the opportunity for a break between the 2, and then also a little bit of a buffer time in case people have problems, technical issues or things along those lines. But I try to gauge it based on the number of questions. So that is how the time is determined for those. Okay.
Okay, thank you.
Any other questions? But yes, my guess is with it being 2 organ systems, there will likely be more questions, which means you will have the more time for the exam. On the exam. Lab, well, both. Probably it'll be probably a longer lab and a longer lecture exam. But I haven't finished writing them yet, so I don't know. All right. Excellent. Have a good weekend. Be safe, be smart. Continue to listen to the doctors. Don't listen to the politicians, although nobody's talking anymore because everybody's apparently fatigued. Apparently, we're just all tired of the COVID. So even though it's going up literally everywhere, nobody cares anymore. So please care. All right. Take care and have a good weekend. See you Tuesday.